Welcome back. This is week 7, lecture 1. So in this week we are going to cover various mega omics projects. So we briefly touched upon it at the end of your week 4, where we said after the human genome project there were various large scale omics projects to cover various aspects of omics. So when we talk about mega omics projects, what distinguishes them from your more localized use of omics is that they are all driven by consortia. So all these mega omics projects, they have different types of consortia. So for example, you could have independent entities. So independent entities could be some industry, or it could be a university, a lab which is located in a university. It could be some government institute which is involved in research. It could be some kind of non-governmental organizations which is again involved in this kind of research. So they are all independent entities. They can of course do omics just on their own. Then these independent entities, the magnitude of data that they can generate and the kind of problems they can tackle is limited by the resources and capacity that these organizations have. But then let's say we combine two organizations. Let's say we combine a university with a government institute. So then you have a very small consortia of two institutes. So of course they pool their resources, they pool their efforts, so then they can tackle slightly bigger problems. Or you could have a consortia of industry and university, so there can be flow of information from these very different entities in how they operate, and then of course you can do other kind of projects. Or you could have two NGOs coming together, or you could even have two industries coming together, two universities coming together. So consortia need not be always between different types of organization. It's just like pooling of resources, whether it is within the same university system or across the university system. And of course you can have slightly bigger consortia where there are more than two entities, maybe 5, 6 entities. And of course you have these kind of mega consortia where there can be multiple entities from very different backgrounds. So there can be many players from industry, there can be many universities involved, there can be a lot of NGOs involved. So these kind of mega consortia also are possible. And of course these kind of consortia can tackle very large problems which maybe these individual entities could not have done on their own. So when we talk about mega omics projects, these are in most cases, right, they are driven by these kind of large consortia. So when we talk about mega omics projects, what kind of projects are we talking about? So for example, you have a genome assembly project. We talked about the human genome project, how there were people from various countries, from hundreds of universities taking part in it. And of course there is more recent projects like the vertebrate genome project, where again there are a lot of stakeholders who have all come together in a consortia, and this vertebrate genomics consortium is working on making genomes for hundreds of vertebrate species, right. So these kind of genome assembly projects, but again at a much larger scale, is one type of genomics or omics projects that is done at a mega scale. You can also have projects to catalog genetic variation. It could be single nucleotide polymorphism, structural variation. It could be characterizing the entire haplotype. For example, we talked about the 1000 genome projects where the goal was to sequence 1000 human genomes. But of course now you have projects which are aiming for larger and larger number of individuals and larger sample size when they are cataloging genetic variation. So of course this is a step that happens after the genome is available. Then the next step probably is to catalog variation across large number of individuals. Then you can also have projects where you are measuring phenotypes and also doing genome sequencing. So then these kind of projects are quite good when you want to do association between the phenotype and the genome. So you want to see, okay, there is a change in the phenotype, then what is the corresponding change in the genome, right? So one example of this would be biobanks, where people are measuring various health parameters of individuals, and also the genomes of all these individuals are being sequenced. Again, this is happening at the scale of hundreds of thousands of genomes or hundreds of thousand patients who are being monitored. So this is another type of mega omics project where hospitals from all over the country could be involved, or a select set of hospitals who have this setup can take part in this kind of consortium. You can also have large scale atlas. Maybe you want to make a tissue atlas, maybe you want to make an atlas of all the cell types that are there, or maybe you want to look at all the cancer types that are there, right? So these kind of projects which plan to build atlas of maybe using a transcriptome or a proteome based approach or a lipidome or any such omics based approach to catalog all the diversity that is there within an organism, whether it is between tissue types or cell types. This can also be another goal of these kind of mega omics projects. So for example, you have this TCGA project, which is a cancer genome atlas, which aims to build atlas of all the different cancer types that are there. So this is one example of cancer type atlas building. Or you have the GTEx project. So in the GTEx project, the goal is to build a tissue expression atlas, that is, you look at different tissues and different individuals, then you see how variable the expression of genes are between individuals and between tissues. So it's a combination of your 1000 genomes project where only the SNPs were being catalogued and gene expression map. So both of them are being done for the same individuals, so that you can look for associations between gene expression change and variation in the genetics. Then of course you can have multi-omic assays, that is, you are not doing just genomics or just transcriptomics or just one or two of them, but you are doing many different assays, whether it is proteomics, epigenomics, again multiple type of transcriptomics, right? You maybe are measuring mRNA, you are measuring microRNA, you are looking at other type of non-coding RNA. So this kind of many omics assays are being done so that the data can be integrated and you can mine for interesting biological information from this. So for example, one example is the ENCODE project, which we touched upon earlier. So it's an encyclopedia of DNA elements. So it started off for the human genome, then of course it has moved on to other non-modeled species, right? Then of course when you talk about consortium, they may focus only on generating large amounts of data, but generally when they generate large amounts of data, they also tend to analyze these diverse data types, because they are the first set of people who get access to such large data sets. So their ability to make very fundamental discoveries is great, because they are the first people who have this resource. And generally these days when we talk about such consortia, they have a very specific goal, right? So if you think about Human Genome Project, the goal was, okay, we're going to make a human genome and then that's going to be a useful resource. But you can have a more specific goal, like you want to find a cure for some specific disease, right? So there may be some disease which is very pressing, urgent matter for a particular group of people. So they can come together, pool resources and try to solve that. So there can be a very specific goal that you generate all this data, you analyze all this data, so that you can achieve this goal of maybe finding a cure for the disease, or at least understanding the biology of this disease in a better way, right? So again, depending on the scope of this project, these mega omics projects can be very different. But the one thing that is common across all these kind of mega omics projects is they tackle very large questions, right, where a lot of resources are required, and they involve such consortia to generate and analyze such large data sets. Right. So when we think about these mega omics projects, are they really worthwhile? Are the outcomes of these kind of projects really useful, and how does it compare to just individual labs doing their own thing, right? So how are they different, or how is the outcome slightly different? So let's say you have mega omics projects, then you can generate genomes of any given set of species that you are interested in. So you don't have to rely upon different groups coming together and somehow putting together all the genomes. You could also generate the transcriptome for large numbers of species. You can also do the proteome. You can also look at the metabolome. You can also do data integration across all these type of data. But if there were individual labs involved, right? So maybe there is one lab which is doing a genome, then there is a different lab which is to do the transcriptome. Maybe it's not doing it now. Maybe it takes them 10 years to generate the transcriptome. Then the proteome is generated by a third entity, and there is no coordination between these entities. So the transcriptome may be of one particular stage or developmental stage or one particular tissue. The proteome may be of a different developmental stage or tissue. So then integrating between the proteome and the transcriptome is almost impossible. Same thing with various other omics assays. So if you want to do data integration, one of the fundamental things that is required is this kind of consortium which have coordinated their efforts so that data integration can be done. And once you can do data integration, of course you can look at overall patterns that are there, you can identify different cell types that are there, different tissue types that are there. You can also see how there is crosstalk between different parts of the biological network. So these kind of data integration can be done when you have this kind of mega omics projects. So what is the outcome? So you could use this kind of data to do biomarker discovery. For example, you want to identify some biomarkers which are a defining feature of some disease, or which are a defining feature of some tissue. So then these kind of data sets which are comparable in quality, which are integrated and comparable in the sense that they represent the same biological state across different assays, they can be very useful when you want to do biomarker discovery, because you can do biomarker discovery in transcriptome and then go and check, are these biomarkers still relevant when we look at the proteome? Or you could do biomarker discovery in the proteome, then go and look at the transcriptome. So then the biomarkers which you discover, you will know whether they are relevant only at the transcriptome level or they are still being produced as proteins. You can also do association studies. For example, you have sequenced the genome, you have sequenced the transcriptome. So if there are some variation in the genome, then does it translate into differences in the transcript expression, and further translated into differences in the protein level, right? So these kind of association studies can be done when the data set is from the, let's say, the same individual, that is the genome, transcriptome and proteome are from the same individual. You can also do novel basic biology, because when you have such a large data set and you can do this kind of data integration, your ability to ask novel questions and identify new things which was not really possible earlier is possible because of this new type of data. It can also help in drug development efforts, because if you are focusing on some specific goal, like some specific disease, so you may have samples where different drugs have been given to patients, and you can monitor the outcomes when these different drugs are given, and this can in turn help decide what type of drugs are effective in this particular disease. So this is again another useful thing that can come out of this, because you have a large enough data set where you can evaluate the effect of different type of drugs. Of course, one of the things that we have seen over the course of all these mega omics projects is, whenever there is one mega omics project and you generate all this data, it seems like, oh, this is such a large project. I mean, this is going to end all these mega omics projects. But generally what happens is, by the end of that mega omics project, the technologies have developed so much, our omic assays have become so standardized, that generally what happens is you end up initiating an even bigger project. Right? So for example, we had the Human Genome Project, which when it started, people thought, you can't have a project bigger than this, you're trying to sequence the whole genome. But by the time the Human Genome Project ended, there were already goals to sequence thousands of genomes, catalog genetic diversity, look at gene expression variation and things like that. Right? So these projects are becoming bigger and bigger by initiating more and more such kind of projects, because of the outcomes that these projects have brought about have been quite useful. You can also have various tools that are developed as part of these omics projects, or you could develop some databases where the general public, means the people outside the consortium, but maybe scientists, maybe general public, can still access it, make sense of it, also provide tools which are very user friendly, so that even somebody who is just a non-scientist who wants to understand some aspects of it can still make use of it. Right? So these kind of tools and databases are also one of the major outcomes of these kind of mega omics projects. So when it comes to data integration, right? So we've been talking about data integration. You can have genomics, epigenetics, transcriptomics, proteomics. You can have all these different assays. But when we say data integration, what exactly do we mean, right? So this term can be used in very different contexts. So for example, when we say data integration, one very simple thing is you can just look for correlations. So you can see if the genomic variation correlates with the epigenetic variation. So is there a positive correlation or is there a negative correlation? So positive correlation could be, okay, you have a certain SNP, then there is a certain epigenetic mark. Right? So this could be a positive correlation, or you could have a negative correlation where you have a genetic SNP in some place. Whenever you have that SNP, the epigenetic mark is missing, right? So that would be a negative correlation. So you can do those kind of pairwise correlations between each of these assays, right? You could correlate genomics and transcriptomics, proteomics and transcriptomics, or you could do correlations across more than 2 entities as well. So this is one very simple way of doing data integration. A slightly more sophisticated way to do data integration is at the network level, right. So whenever we talk about biology, of course biology is organized in the form of biological networks where different genes or proteins interact with each other, and this gives rise to complex biology. So now if you want to identify cause effect relationships, that is, okay, if there is some change in this particular part of the biological network, this is going to have this particular effect, right. So this kind of a cause effect relationship could be very interesting. For example, if these genes, right, the red ones, if they are highly expressed, then the genes which are in blue, they are probably going to be downregulated. So this is some kind of a cause effect relationship. So now if you are to just do this using just transcriptomics or just epigenetics, your ability to infer the cause effect relationship may be limited because you are only looking at part of the data, right. But if you have all these different assays, so then you will know exactly how the cause effect relationship is being transferred from one particular protein or one particular gene to another. So you can better integrate the data at a network level if you have multiple such omics assays. Then this is like what people have been doing, but then all of this is based on what we already know about the biology, right. So we already know that, okay, there can be correlation between gene expression and epigenetic marks, or we know that there is a relationship between network X and network Y in the biological networks, right. So then these kind of data integration will work. But what if you don't know what are the relationships that are there between these different assays, but you want to pick up new relationships that are there in the data but which you are not aware. So that's where the use of machine learning kind of tools becomes very relevant. So here you try to infer either indirect, that is they may work through a series of entities which are not known. So it could be that this particular gene acts on this particular gene, then this acts on this gene, then this acts on this, then there could be a very indirect relationship, and that finally ends up affecting this gene, right. So this may not be obvious just by the biology that has been identified or that is known to us till now, or this could be some kind of a novel relationship. So this actually makes sense, it's just that we have not yet identified it, right. So these kind of relationships can be inferred using these kind of machine learning tools, which when they integrate across these data, they can pick up such signals which was not obvious to us earlier, right. So these are some of the examples of how you can do data integration when you have these kind of very different assays. So one of the things that has become possible thanks to these mega omics projects is more and more sophisticated ways to do data integration are becoming available, not just for those omics projects but to the overall scientific community. So one example of data integration, or where you have these kind of large kind of data sets where you have different assays, right. So you could have gene expression, you could have ChIP-seq, you could have chromatin interaction profiles, right, how different parts of the chromosome are interacting. So you can have many such assays. You could also have gene expression, you could have SNPs, right. You have many such assays, and you also have this across many individuals or many different cell lines, right. So this, for example, is DNA modifications that are there in one individual. This is in second individual, third individual, fourth individual. You have histone modifications. Again, these could vary between individuals. You can have histone variants. You can have nucleosome occupancy. There could be RNA modifications, that is some changes to the RNA, or there could be some non-coding RNAs, how they are expressed. You can have chromatin interactions, then the chromatin domains, right. So something like your topologically associated domains, right. So these kind of assays can be done, and they can of course be done across different tissues, cell types, or experimental conditions. So they could also be different individuals. So then you have these kind of data across many individuals and across many assays. So it's a multi-dimensional type of data. So when you have this kind of multi-dimensional type of data, one thing that can happen is there can be a lot of missing data. So for example, when you're doing all these assays, maybe in this particular individual, let's say we call it individual 2, the information is not available for some assay. Either the assay failed or the quality of the assay is not good. So then you end up having some missing data. So then when you have this kind of missing data, the solution would be that you impute it. That is, given the information that is available for the other individuals, and the information available in the other assays, you can infer what probably is the state of this particular data in this particular sample. So, this kind of imputation of data, whether it is across such multi-dimensional data set or even some simpler data set like SNP data set, is quite common when you have this kind of large data sets, when you have enough statistical power to make such predictions. Another thing that you can do when in terms of data integration is aggregation. So, when we say aggregation, let us say you have all these different data types and you have them across many individuals. Of course, you can impute and fill up some of these data, but then if you look at all these data, it is very difficult to infer what they mean overall, right? Because if you just look at histone modification, maybe it makes sense, maybe you just look at nucleosome occupancy, it makes sense. But if you want to combine across them, that is this combination of histone modification, this combination of nucleosome occupancy and this particular set of chromatin domains, what does it mean, or does it have any meaning, right? So, this aggregation across assays and across individuals can be quite useful. So, when we want to do that kind of aggregation, one common tool that has become used is this kind of definition of chromatin states. So, what do we mean by chromatin states? So, you look at all these assays and a particular combination of values, that is this gene is expressed, this chromatin state exists, and this particular interaction is happening between different parts of the chromosome. This combination of states is given one particular mega state, right. So, this becomes a new chromatin state. Then same thing you can say, okay, then this second combination of chromatin states is another chromatin state, right. So, you can aggregate across these different assays and you can define your own new chromatin states using some tools, and then you can see, okay, given this combination of assays and when we have defined this chromatin state, what does it mean biologically, right. So, aggregation is again another very useful way to integrate different data types, and that can give you some new insight when you try to associate this state with some specific phenotype, right. So, now before we go into various examples of mega omics project, let us go back to our HGP or Human Genome Project. So, because this is the beginning of all these consortium projects. So, of course, there were some smaller consortium before the Human Genome Project, but in many ways the Human Genome Project provides a model for how various consortium projects have developed over the years, and again it ran for more than a decade, right. So, the 90s way into the 2000s, and there was both public and private contribution to the Human Genome Projects. Of course, the Cellra Genomics, which we talked about in the very early lectures, was the company that was a competitor but then became part of the project because the efforts were pooled. And within the public initiative there was the NIH, then of course the Department of Energy, and there were various sources of funding that was coming into this, and then there were various entities that were controlling how the project was coordinated. And then finally everything came under the umbrella of the NHGRI, which is the Genome Research Institute, which provided a common place for various universities, whether it is in the US or in other countries, whether it is UK, China, various other partners that were there in this international effort. All of them came together in this. Of course, there were some entities where there was an independent effort happening within the public sector as well. For example, in the JGI, Joint Genome Institute, where some of the efforts were happening in parallel. So, this is the structure of your Human Genome Project consortium, where there are multiple entities, some are universities, some are industries, some are NGOs, and again they are in different countries. All of them are put together and then they try and solve this challenge of sequencing, the Human Genome Project. So, when we talk about this, I guess the one interesting example which I thought we should probably discuss is that China actually took part in the Human Genome Project. So, why is this so interesting is at the time when China became part of the Human Genome Project, China was not really this kind of having this large economy that we see today. It was still very early days for China to pick up the growth engine, and you could say that it's a developing country where resources were limited and things like that. So, it makes a good comparison as to how the genomics era of 2 decades, 3 decades, how it has changed as China has changed. Right? So this is something actually from a paper which discusses about the effort, Chinese effort. So what they say is, let's say there is one day a pupil asks such a question in class. So this is an imaginary situation that this person conjures up, and this is what the question that's being asked by the student to the teacher. So what they're asking is, why didn't China do something for the HGP? Right? So why didn't China take part in the Human Genome Project? So the teacher has a reply, right? So again the reply is complex. So what the teacher is saying is, he would have deeply regretted if the answer could only have been the following, right? Of course this was not the case, because of course China took part in the Human Genome Project and it did its part, but if it had not done that part, if it had not taken part in the Human Genome Project, the answer would have been something like this, right? So it would have gone something like, country was very poor, people were illiterate, decision makers were short-sighted, scientists were disappointing. So all of these reasons why China, although everybody of course considers the country great, didn't actually become part of the HGP, and we lost out on this opportunity, right? But then it ends. Of course this will never come true, because China decided to take part in the Human Genome Project and do its part, right? So this again it says, okay, it's written by Chinese middle school teacher who was asked this hypothetical question and this hypothetical answer. But this actually portrays the situation that was there in China, and how the entry of China into this was a big event, because this changed the direction of the Human Genome Project and genomics in China. So this again taken from this paper where the China's contribution to the HGP is discussed in detail. So this is the Chinese team joining the 5th strategic meeting of the HGP in the UK. So you see there are some Chinese scientists who are taking part in this meeting, and this is the first time China is becoming part of the HGP. So what exactly did China do? So they actually managed to sequence 1% of the human genome. So this part is also known as the Beijing region. So if you go on chromosome 3 of the human genome, at the very end of the chromosome 3, right, in the P arm, there is this region here. This region is known as the Beijing region or 1% project. So if you actually zoom in and you see what are the different regions that have been sequenced. So you see there are various accession numbers which have been labeled as Beijing region, right? So these are the artificial chromosomes that China actually managed to sequence. It's a very small part of the genome, right? It's just only 1% of the Human Genome Project, but this whole thing is done by China, right? And this gave them really a flavor for how the Human Genome Project was happening and how they can contribute to it and also learn from it. So this actually some of the initial things that happened in China. So of course, as I was saying, China was still very early in its growth story. So if you see the lab that I've built, it's efficient and economic lab, because the benches that are there, they're all built out of cardboard boxes, right? So this just goes on to tell you how frugal the effort was, and really trying to save up on money. Of course, there were all these sequencers that are placed here. So these are all Sanger sequencers where the sequencing was done, how they could sequence this 1% of the genome. And of course, there was a lot of effort involved and a lot of money of course was spent to give you an idea of how much effort was there. So this is actually a box which they had kept in this Beijing region sequencing project, where all the tips, right, the pipette tips which were used were collected in these huge boxes, right? So whatever pipette tips were used for pipetting for various steps in the sequencing, they've all been collected in these boxes. So there are 15 million tips that were used for 500,000 sequencing reactions, right? So that's around 30 reactions in 6 months. So this is actually done in BGI from October 99 to April 2000. So this is the magnitude of the effort, where sequencing is done at a very large scale, and the goal is sequencing 1% of the human genome, right? So this how China really got involved, and as we saw earlier, now of course China has come to a stage where BGI is developing its own sequencing platform and sequencing large numbers of genomes very routinely. So this gives you idea how new entrant can come into a consortium and really change how science is done within the country or within that particular entity and learn from this bigger consortium. Right, so let's wrap up. So of course there are many advantages like this for the consortium mode projects. So for example, you pool resources, then you can use the resources optimally, you can plan how the efforts are distributed, so you can assign tasks which somebody is good at, and you can take advantage of the expertise of different individuals or entities that are involved. And main thing is you can avoid duplication of efforts, right? So if the Human Genome Project was not coordinated, right, if China was sequencing human genome separately, they're not sharing anything, the US is sequencing the human genome separately, UK is separately sequencing, so they would have to sequence the entire human genome all on their own, right? So then they would be duplicating all that effort to sequence the same thing. But since the project was well planned and there was this international initiative, they could decide, okay, China is going to sequence this part, right? So chromosome 3, this 1% is going to be sequenced by China, right? And then nobody is going to duplicate that effort. So this was possible. Then when you have these kind of large scale efforts, you can really answer broader questions, because if the Human Genome Project was not really coordinated in this way, if different labs were just sequencing some random parts of the human genome, of course there would be a lot of duplication of effort, but also some of the questions that could be asked once the whole genome was available could not be asked, if the data was distributed all over the country, right, or all over the world. Only because the whole data set was assembled into one large human genome, you could look at various questions which otherwise would not have been possible to answer. Then another thing which was probably the most important thing or most important outcome of the Human Genome Project was various tools and benchmarks were developed, whether it be the next generation sequencing revolution that happened, or various sequencing formats like FASTQ or variant call format. All of these got standardized since everybody was working together and coordinating their efforts. So they needed a way to share data across groups, share data across the consortium. So this would have only been possible if there was some standardized tools. But imagine if this was done, without any coordination. Each group is proposing their own file format, each group is sharing data in a different format. Just converting the data from one format to another just to get it all into one format would be a nightmare, right? So again, this is overcome because this is working in a consortium. And since you are a consortium, you can cross-validate results, right? So you could take parts of the data that have been generated by a different entity in the consortium, and you can validate that in another lab in the same consortium. You can see, okay, how good or how reliable is the quality when it is coming from different entities in the consortium. So this can ensure a high quality of final output, right? Because if your individual labs are doing this, so there is no way to evaluate the quality. You would have to go and replicate the whole thing. But if there is cooperation, then there can be cross validation of these results. So this can ensure higher quality of the output. Then another thing is you have these entities from industry, academia, all coming together. So there are a lot of opportunities for networking because people can then move from academia to industry and the other way around. So there aren't these artificial barriers because they are all working under this big umbrella project, and there is always networking going on between them, right? So these are some of the advantages when it comes to your consortium type of projects. But this is not to say that there are no challenges associated with consortium mode projects. Of course there are, right? For example, we said that data quality can be maintained, right? We can have higher quality, but generally this requires extensive centrally monitored QC protocols. That is, you need some central governing body which ensures that all these entities adhere to certain protocols and they actually are generating data in that quality. So there can be this extra effort where there is central monitoring required. Then when we are saying, okay, these consortia are working for the common good, of course, yes, they want to do sharing of data, interpretation, publishing as a consortium, all of this is there, but of course this needs careful planning because different entities involved in this project may have conflicting interests. So somebody may be interested in some particular part of the genome, they have some genes of interest in that, so they want to publish their results and they don't really care about the biology of other parts. So there can be very different such conflicting interests of the entities. So there needs to be good coordination so that these kind of conflicts of interests are managed. Then of course when you have contribution from such diverse partners, it can be very tricky to assign what is the contribution of individual entities. For example, in the Human Genome Project, we saw that there is 1% of the human genome is sequenced by China. But then the rest of the 99%, how much of it is sequenced by the US, how much of it is sequenced by the private enterprise, how much of it is sequenced by the public enterprise, how much was sequenced by both of them very close by, right? All of this is very tricky to quantify because this happens very fast, and then sharing credit can be tricky when you do these kind of consortium mode projects. And when you work in these consortium mode projects, again the individual goals of the entities may not align. For example, you have the public and private enterprises involved in the Human Genome Project. So the private enterprise, the goal was to patent it, make money out of the Human Genome Project, because of course they are driven by their shareholder needs, right? But the public initiative had a very different goal where they wanted to make the data available to everybody free of cost. So the individual goals of these entities are not really aligning, but then when you work in a consortium, you need to somehow figure out how best to make these things align. Then after you have generated whatever data resource you are generating or whatever novel biology you are discovering, patenting of these results and how the IP is shared can be very challenging again when the goals are very different. But then you also have this thing which is fun of scientific discovery, right? So it's what a driving force for most scientists, that's the reason that they want to do science. This could be lost, right? Because if you are doing these kind of huge mega consortium projects, you're just given part of the task and you're like a part in this huge production line or a cog in this big machine, and then you're just doing your part, right? Then of course you will end up doing some scientific discovery, of course you will end up finding some new things, but the fun of making scientific discovery may not be the same when you work in a consortium versus when you're doing it individually. So of course this can be very different in a philosophical sense compared to your non-consortium projects. Right, so we'll end this lecture here and continue with some examples of consortium projects in the next lecture.